For most of recorded history, the story of plague was told in 3 chapters, each one a documented catastrophe with a name, a date, and a body count. The Plague of Justinian, striking the Byzantine Empire in 541 CE. The Black Death, killing somewhere between a third and half of Europe's population during the 1300s. The Third Pandemic, beginning in China during the 1850s, and eventually spreading by ship to port cities across the globe. 3 outbreaks, roughly 8 centuries apart, treated for generations as the complete story of humanity's relationship with one of the deadliest bacteria ever to infect our species. According to ancient DNA, that story was missing an enormous chunk of its own beginning. Buried in the bones of hunter-gatherers who lived and died thousands of years before the pyramids of Egypt were built, researchers have found direct genetic proof that Yersinia pestis, the bacterium responsible for plague, was already infecting and very likely killing human beings roughly 5,500 years before its first documented outbreak. This isn't a minor adjustment to an established timeline. It pushes the story of plague out of the realm of recorded history entirely and into the deep, undocumented world of prehistoric Europe and Asia, forcing researchers to rethink not just when plague began, but what role it may have quietly played in some of the biggest population changes in human prehistory. To understand how significant this correction actually is, it helps to walk through what researchers believed before ancient DNA sequencing made this discovery possible. For most of the 20th century, the study of plague's deep history relied almost entirely on written historical records and, further back, on informed speculation. Since bacteria don't fossilize the way bones do, and since Yersinia pestis leaves no obvious visible mark on skeletal remains the way some diseases do, there was simply no direct physical evidence available to search for cases predating written history. The Justinian plague, documented by Byzantine chroniclers in considerable detail, was treated as the earliest confirmed pandemic in the historical record, with anything before that period existing purely as an open question. Historians and epidemiologists could speculate about but never actually confirm. This changed dramatically once researchers developed the ability to extract and sequence ancient pathogen DNA directly from the teeth of long-dead individuals. Dental pulp, protected deep inside the tooth by a layer of enamel, turns out to be a remarkably effective place for genetic material to survive for thousands of years, including genetic material from pathogens that were circulating in a person's bloodstream at the time of their death. This technique opened up an entirely new way of investigating ancient disease, one that didn't depend on written records existing at all. If you find it fascinating how much of ancient history is quite literally locked inside our teeth, waiting for the right technology to unlock it, that's exactly the kind of discovery playing out here. Go ahead and hit that like button, because what researchers found once they started applying this technique to prehistoric burials was considerably more dramatic than anyone initially expected. In 2015, a team of researchers published findings from a large-scale screening effort testing DNA extracted from 93 Bronze Age and Stone Age human skeletons recovered from burial sites scattered across Europe and Asia. Among these ancient remains, several individuals tested positive for Yersinia pestis, with the oldest confirmed cases dating back approximately 5,000 years, placing confirmed plague infection firmly within the late Neolithic period, roughly 3,000 years before the birth of Christ, and well over 3,000 years before the Justinian plague, the previously recognized starting point of documented plague history. Let that sink in for a moment, because the scale of this correction is genuinely enormous. This wasn't simply a matter of pushing the timeline back a few centuries based on a newly discovered historical text or a slightly earlier written account. Ancient DNA evidence demonstrated that plague was already circulating among human populations for literally thousands of years before anyone ever wrote the word down, an entire unrecorded chapter of the disease's history that traditional historical research had no way of ever accessing. Even more striking, subsequent research pushed this timeline back further still. A 2021 study identified an even older confirmed case, extracted from the remains of a hunter-gatherer individual, one whose genetic profile placed the infection close to the very root of the entire Yersinia pestis family tree, essentially capturing a snapshot of the bacterium near the earliest point in its evolutionary history that researchers have directly observed, combined with molecular clock estimates, which calculate roughly how long ago 2 genetic lineages diverged based on the rate at which mutations accumulate over time. This evidence suggests Yersinia pestis itself may have first evolved somewhere around 7,000 years ago, meaning the bacterium had already been infecting human populations for a solid 2,000 years before the specific case captured in this ancient hunter-gatherer's remains. If uncovering these hidden, undocumented chapters buried directly inside ancient teeth and bones fascinates you, make sure you're subscribed, because ancient pathogen DNA keeps rewriting timelines exactly like this one across the entire history of infectious disease. It's worth walking through exactly how researchers managed to screen for a specific ancient pathogen among a large collection of skeletal remains, since the methodology itself explains why this kind of discovery took as long as it did to make. Researchers extract genetic material from the inner pulp chamber of ancient teeth, then run that extracted material through a process that compares it against a reference database containing genetic signatures from thousands of known modern pathogens. Because the vast majority of genetic material recovered from an ancient burial belongs to the deceased individual themselves, along with substantial contamination from soil bacteria and other environmental sources, identifying a specific pathogen buried within. That noise requires extremely sensitive computational screening techniques capable of picking out a comparatively tiny signal from an overwhelming amount of background genetic material. This entire approach only became feasible relatively recently. The sequencing costs dropped dramatically, and computational screening tools became sophisticated enough to reliably distinguish authentic ancient pathogen DNA from contamination or statistical noise. Before these tools existed, ancient remains could be examined endlessly using traditional archaeological and skeletal analysis without ever revealing the pathogens that may have been circulating in an individual's bloodstream at the time of death. Since bacterial infections, including plague, typically leave no distinctive mark on bone at all. Here's where the story becomes genuinely fascinating from a scientific standpoint, because these earliest confirmed cases of plague turn out to look meaningfully different from the disease responsible for the Black Death and other historically documented pandemics. Modern bubonic plague spreads primarily through fleas, which pick up the bacterium after biting an infected rodent and then transmit it to humans through subsequent bites, a transmission pathway that depends on a specific gene, commonly referred to as the YMT gene, which allows the bacterium to survive and multiply inside a flea's digestive system long enough to be transmitted effectively. When researchers examined the genome of these earliest known plague cases, they discovered that many of these ancient strains lacked this crucial gene entirely. Without it, the bacterium would have had considerable difficulty surviving inside fleas long enough to spread the way bubonic plague famously did during later historical pandemics. This suggests these earliest documented infections likely spread through entirely different pathways, potentially through direct respiratory transmission between infected individuals producing a pneumonic form of the disease, or through direct contact with infected blood or tissue causing a septicemic form. Both of these alternate forms of plague are, if anything, even more immediately dangerous than the bubonic form most people associate with the Black Death, though they typically spread less explosively across large populations, since they generally require a more direct contact between infected individuals rather than an intermediate flea vector capable of carrying the disease across considerable distances. This genetic detail matters enormously for understanding how the disease's evolutionary history actually unfolded. Rather than emerging in its most famous flea-transmitted form and remaining essentially unchanged across thousands of years, Yersinia pestis appears to have evolved considerably over time, gradually acquiring the specific genetic adaptations that eventually allowed it to exploit fleas as an efficient transmission vector, a development that dramatically increased its capacity to spread rapidly across large human populations. The devastating, fast-moving pandemics that shaped medieval history represent a considerably more evolved, more transmissible version of a bacterium that had already been quietly infecting human populations in a somewhat different and likely less explosively contagious form for thousands of years beforehand. This evolutionary picture has also grown more detailed as researchers have recovered additional ancient genomes from different time periods and different regions, rather than a single, clean, linear progression from an early non-flea adapted form directly into the fully flea-transmissible version responsible for later pandemics. The accumulating genetic evidence suggests something closer to a branching family tree, with multiple distinct lineages of Yersinia pestis emerging, spreading, and in some cases apparently disappearing again at different points across several thousand years of prehistory. Some of these ancient lineages appear to represent evolutionary dead ends, strains that circulated for a period among specific populations before vanishing from the genetic record entirely, while others show clearer connections to the specific lineage that eventually developed the genetic adaptations behind the Black Death and its associated pandemics centuries later. Mapping out exactly how these different ancient lineages relate to each other and which specific branch eventually gave rise to the medieval pandemic strain remains an active area of ongoing research, with new ancient genomes occasionally reshuffling researchers' understanding of exactly how these different branches connect. This raises a genuinely significant question that researchers are still actively working to answer. If plague existed for thousands of years before it evolved the specific genetic adaptations needed to trigger the kind of explosive, society-altering pandemics documented in written history, what impact, if any, did these earlier, less transmissible forms of the disease actually have on prehistoric populations? This question connects to one of the more intriguing and still actively debated ideas in current archaeological research, sometimes referred to as the Neolithic decline hypothesis. Archaeologists have long puzzled over a genuine mystery embedded in the European archaeological record, a period roughly 5,500 to 5,300 years ago, when many large, densely populated Neolithic farming settlements across parts of Central and Eastern Europe appear to have been rapidly abandoned, with population density dropping sharply across a relatively short span of time. Traditional explanations for this decline have included climate change, soil exhaustion from intensive farming, and social or economic instability. The discovery of ancient plague DNA, dating to almost exactly the same period, has led some researchers to propose an additional contributing factor worth taking seriously: the possibility that early pre-flea adapted strains of plague may have swept through these large, densely packed farming communities that would have been particularly vulnerable to a disease capable of spreading through close respiratory contact, given how tightly concentrated their populations were compared to smaller, more dispersed hunter-gatherer groups. It's important to be careful and precise about exactly what this evidence does and doesn't establish, because this particular hypothesis remains genuinely debated within the research community rather than settled fact. Finding ancient plague DNA in skeletal remains from this general period confirms that the disease was present and circulating among these populations. It does not, on its own, prove that plague was the primary or even a significant driver of the broader Neolithic population decline, since correlation in timing doesn't automatically establish a direct causal relationship. Some researchers studying this question remain skeptical, pointing out that confirmed plague cases from this period remain relatively few in number given the scale of population decline being discussed, and arguing that climate and agricultural factors likely deserve more weight in explaining this specific archaeological pattern. Other researchers argue that plague deserves serious consideration as at least a contributing factor, particularly given how well the timing aligns and how vulnerable large, dense Neolithic settlements would have been to a newly emerging respiratory pathogen. This is exactly the kind of genuine, ongoing scientific disagreement that reflects healthy scientific debate, rather than any kind of weakness in the underlying research. Different teams weighing the same core evidence differently while further data continues to accumulate. If you're the kind of person who enjoys watching an open scientific debate play out in real time, rather than only hearing about settled conclusions after the fact, this is a genuinely good example to follow, since new ancient DNA samples continue to be analyzed and published on a regular basis, gradually adding more data points to help researchers eventually resolve exactly how significant the role early plague strains actually played during this specific chapter of prehistoric Europe. It's also worth noting how this deep timeline connects to plague's more recent, better documented chapters, since the contrast helps put the scale of this discovery into perspective. The third pandemic, beginning in China's Yunnan province during the 1850s before spreading globally through shipping routes, is the best genetically documented plague outbreak in history, having occurred recently enough that researchers have access to extensive written records, detailed case counts, and even preserved bacterial samples from the period itself. Comparing the genome of this well-documented modern outbreak against the ancient Bronze Age and Neolithic genomes recovered from prehistoric remains allows researchers to calibrate their molecular clock estimates with considerably more precision than would otherwise be possible, essentially using the well-understood recent history of the disease as a known reference point to help interpret the much older, far less certain genetic signals recovered from ancient teeth. This kind of cross-referencing between recent, well-documented outbreaks and ancient, undocumented ones is exactly what allows researchers to speak with reasonable confidence about events occurring thousands of years before any written record existed anywhere in the world. There's another layer of this story worth exploring, because the geographic and genetic distribution of these ancient plague strains connects to one of the biggest population movements in European prehistory, the migration of steppe pastoralists associated with the Yamnaya culture, who began moving westward from the Pontic-Caspian steppe into Europe starting around 5,000 years ago. Several of the oldest confirmed ancient plague genomes have been recovered from individuals connected to this exact migratory movement, raising an intriguing possibility that these expanding steppe populations may have carried early strains of plague along with them as they spread across the continent, potentially introducing the disease to new regions and new populations who had never previously encountered it. This wouldn't necessarily have looked anything like a dramatic, fast-moving epidemic in the way we typically imagine historical plague outbreaks. Given the likely absence of efficient flea-based transmission at this early stage, spread may have been considerably slower and more localized, occurring through direct contact within small, close-knit groups rather, than exploding rapidly across entire regions the way the Black Death famously did many thousands of years later. Taken together, this expanding body of ancient DNA evidence paints a picture of plague's history considerably longer, considerably more evolutionarily complex, and considerably more intertwined with major prehistoric population movements than the traditional three-pandemic framework ever suggested, rather than a disease that emerged suddenly during the Byzantine era, wreaked catastrophic havoc across 3 distinct historical episodes, and then largely faded into modern medical irrelevance following the development of antibiotics. Plague now appears to represent one of humanity's oldest and most persistent bacterial companions, one that has been gradually evolving alongside us, adapting new transmission strategies over thousands of years, long before any human society developed writing systems capable of documenting its presence. This broader recognition carries an important lesson about the limits of relying purely on written historical records to understand the deep history of infectious disease. Written records, however detailed and however carefully preserved, can only tell us about diseases significant enough to be noticed, named, and documented by the specific societies that happen to develop writing systems capable of recording them. Ancient DNA research operates under no such limitation. It can identify a pathogen's presence directly from skeletal remains, regardless of whether the society in question left behind any written record at all, offering researchers a genuinely unprecedented window into disease history stretching back tens of thousands of years before the earliest known writing systems ever developed. This has profound implications, extending well beyond plague specifically. If a pathogen as significant and as well studied as Yersinia pestis was quietly circulating among human populations for thousands of years before its first documented historical outbreak, it raises a genuinely important question about how many other major pathogens throughout human history might have similarly ancient, currently undocumented origins, simply waiting for the right ancient DNA sample and the right sequencing technology to finally reveal them. Researchers studying ancient disease increasingly suspect that plague is unlikely to be a unique case, and ongoing ancient DNA research continues actively searching for evidence of other pathogens with similarly deep, previously unsuspected prehistoric roots. There's something genuinely humbling worth sitting with here, a pattern that keeps repeating itself across so many different corners of ancient DNA research. For generations, entire fields of study confidently built their understanding of the past around the specific evidence available at the time, whether that evidence came from written historical records, skeletal analysis, or archaeological excavation. That research wasn't careless or unserious. It represented the most complete and reasonable interpretation available using the tools and evidence accessible at the time. But written records and skeletal analysis alone, no matter how carefully studied, simply cannot detect an ancient pathogen's presence the way direct genetic sequencing now can, and that fundamental limitation left an enormous multi-thousand-year gap in our understanding of plague's actual history, one that remained essentially invisible until the right genetic technique finally arrived to fill it in. So the next time plague comes up in conversation, framed as a medieval disease that struck fast, killed millions, and then eventually receded into modern medical history following 3 dramatic historical episodes, it's worth remembering what the ancient genetic record actually reveals instead. Long before Byzantine chroniclers ever wrote about plague sweeping through Justinian's empire, long before any pyramid was built in Egypt, and long before agriculture had even fully taken hold across large parts of Europe, this same bacterium was already present, already evolving, and quite possibly already playing a quiet, largely invisible role in shaping which prehistoric communities thrived and which ones didn't. Plague isn't simply an old disease. According to the deep genetic record now being pieced together one ancient tooth at a time, it may be one of the oldest, continuously evolving threats our species has ever faced, quietly present in human bloodstreams for thousands of years before any of us ever thought to give it a name.